those work. We work on natural resource and agricultural issues that are important to the tribe, or all the tribes. And I'd like to take this time to thank Andy, Michael, Carla, and Sarah for this, and all of the planning team for putting this forum together. It's those one of those things that's like mixed feelings. It's a great topic, but it's a depressing topic. But it's so exciting, the quality, the expertise of the presenters. So that's really the big thing, that when just being involved in this, to see the quality of the speakers. So this next section will be the impacts of neonics on biota. And our first presenter is going to be, we have two presenters from the United States Geological Survey. The first one is Dr. Samatha Oliver. She's a hydrologist with the USGS, the Upper Midwest Water Science Center. Our second presenter is Dr. Travis Schmidt from the USGS. He's with the Wyoming-Montana Water Science Center. And then we have Shawn Giblin, the Mississippi River Water Quality Specialist with the Wisconsin Department of Natural Resources. We'll have this the same way, each presenter to come up, and then all the speakers come up at the end for questions. So if we're ready to get started, it's all yours.
Thanks, Jeff, and thanks everyone for having me today. As he said, I'm Sam Oliver. I'm with the U.S. Geological Survey. I'm located here in Madison in the Upper Midwest Water Science Center. And you'll notice that the words neonics and Wisconsin are not in my title, so I apologize, but I'm also here to provide context, how neonics are just part of the overall pesticide picture, and I'll also give you context, for sort of comparing Wisconsin to what's going on in the greater Great Lakes region. So first I want to say that all of this work is published in 2 papers, so if you are interested in the nitty-gritty details about what we did, feel free to go to these 2 resources. And I also want to thank all of my co-authors who contributed a lot to this work. Okay, so the Great Lakes Restoration Initiative funded this body of work, so I just wanted to say a little bit about the context that we're in here. The Great Lakes are home to around 35 million people, and there's a huge gradient in how the land that drains into the Great Lakes is used. We have really dense urban areas like Milwaukee and Chicago. We have high intensity agricultural systems, but we also have pristine systems that drain into Lake Superior, for example, that are mostly forested or wetlands. And the Great Lakes Restoration Initiative started in about 2010. It's funded by Congress. And the goal of GLRI is to restore and protect the largest freshwater system in the world. And so, of course, one of their interests is in emerging contaminants in the Great Lakes Basin. And so this is a map showing all of the sites where we have measured emerging contaminants in the Great Lakes Basin. So we know a lot about what's out there, and there is a lot out there. And GLRI specifically is interested in understanding where do we expect potential negative impacts from these chemicals in the environment, and which of those chemicals should we be most concerned about? Because we all know there are thousands of compounds out there in the environment. We can't do everything. Which ones should we focus on? So that's sort of the Great Lakes and GLRI context that I'm coming from. In phase 1 of GLRI, there was a broad survey of compounds trying to answer these questions: which compounds should we be concerned about? And that led them to 3 chemical compound sort of classes that they were interested in following up on: pesticides, PAHs, and pharmaceuticals. And so this work was born out of that original survey work about which compounds we should be concerned about. And so in phase 2 in 2016, we studied pesticides in the Great Lakes tributaries. So what were we interested in knowing about pesticides in Great Lakes? We wanted to know what and where. What pesticides occur in streams that drain into the Great Lakes, and which sites should we be concerned about? How does the presence of pesticides relate to how that land is being used? And we also wanted to know the seasonality of these compounds. Of course, we associate pesticide use with the growing season and agricultural systems, but we've heard a lot of evidence today that suggests these compounds stick around, they accumulate, they break, have different rates of breaking down. And so we wanted to know from a stream biota perspective, When are these compounds expected to have negative impacts? Is this, a few months of the year, or is it year-round? So when we're trying to evaluate negative impacts to aquatic communities, we're starting at pesticides being applied on the landscape. And you've already seen these maps quite a bit today. This is an example of imidacloprid from 2016, the same year that the study started. We can use maps like that to guess which streams will be impacted by, say, imidacloprid. But just because something is applied doesn't mean it's going to have impact in the stream. There are a lot of processes that happen between application and a chemical, causing a negative impact in the stream. Many of those you've heard about today. There are many that you won't hear about today. It's complicated, I guess, is my point. So once, the pesticide is applied on the landscape, it takes a lot to get that pesticide to the stream. We heard about it taking 20 years in some cases. So if you think about how it gets to the water, those compounds are transforming, they're degrading. Whether they're water soluble or not matters for whether or not it makes it into the stream. Once it makes it into the stream, we still don't know how it's impacting communities. There's a lot that goes on between these two endpoints. The concentration, we've heard a lot about concentrations today, matter. How the organism is being exposed to the chemical, whether they're directly consuming it, absorbing it through their gills. All of those pathways matter to how the organism is experiencing the chemical. The toxicity of the compound matters, the mode of action, what it's actually doing to the biology. And then if there are multiple stressors, we're just talking about neonics today. I'm going to tell you there are a lot of pesticides out there, but there are a lot of other things impacting organisms. If the stream temperature is warming, it's exceeding some temperature threshold, the organism is already stressed, and then it's exposed to pesticides. You can imagine that those stressors can interact. And then, of course, we've heard a little bit about this, but the community structure, who's eating what matters in terms of the overall impact to the ecosystem. So it's complicated. What did we do? We measured 232 pesticide compounds in 16 tributaries of the Great Lakes. And these weren't random samples, these are random sites. We specifically selected these sites to capture a gradient of land use and land cover, from really pristine systems in the Bad River and the St. Louis Estuary River up that drains into Lake Superior, all the way to highly urbanized systems like the city of Milwaukee, to really row crop agriculture dominated watersheds. So we were really looking across this spectrum of sites. And we also captured that annual dynamic by taking monthly surface water samples. So once a month, each of these sites were sampled for the most part. So just to orient you back to this, really complicated diagram, what are we measuring and what can we say with that information? We're measuring directly concentrations in the stream, so we know which pesticides are there. But then we have to put those concentrations in context of how they might impact biology. And you've heard a lot about that today, so I'm not going to go into a lot of detail. What you've heard a lot about are these aquatic life benchmarks, which are kind of the gold standard for how we evaluate these compounds, where organisms have been tested in a lab, exposed to a compound, and we measure, how many of them die. There's another source of information that we used in this study, which is called the ToxCast database. And this is called an in vitro assay. It takes place outside of the organism. So this is like taking cells, exposing it to a compound, and measuring the response of those cells. There are drawbacks to this approach, but it's relatively inexpensive compared to whole organism studies. And so generally it allows us to have a lot more information about a lot more compounds. The drawback is we don't know how that directly relates to how the compound is impacting an animal in the stream. But it's still information, and I want to emphasize that this study was a surveillance study, and we are trying to estimate potential negative impacts. So it's a screening tool, and that's how we're using it here. I won't say a lot about this, but we did evaluate kind of mixtures of pesticides and how those might be impacting organisms. Okay, so I'm going to go through some results here to give you some context of what this picture of pesticides looks like across the Great Lakes Basin. In a lot of my figures, I've just highlighted the Wisconsin sites, so you can kind of pick those out. And then in various slides, I've also pointed out the Neonics just to kind of orient us. We found pesticides everywhere. In 190 out of 198 samples, we detected at least one compound, and pesticides were detected at all 16 sites. Of all of those 232 compounds, we detected almost half of them, 104 were detected. And getting back to this like mixtures of compounds concept, multiple stressors concept, in 80% of all of the samples, 10 or more pesticide compounds were detected. So when we're considering impacts to biota, we should be, thinking about this collective multiple stressors type approach. When are these pesticides present? I'm showing you on the top the concentration, like the summed concentration. So the box plot is representing, variability across the sites. And then on the bottom, I'm showing you the number of pesticides detected. Yes, we see elevated concentrations and number of pesticides detected during the growing season. You can see, May, June, July, August, higher concentrations, higher number of detected compounds. But we're seeing both concentrations, detections, and the number of compounds elevated throughout the entire year. So pesticides aren't a problem just in the growing season for aquatic biota. Which tributaries are sort of the worst off in considering potential biological impacts of pesticides? This figure is a little bit complicated, but first on the right-hand side, I'm showing you what the land use and land cover of each of those sites is. So the purple is really urban, and I've kind of classified the sites as urban on the top, row crop dominated kind of in the middle, ag mix, which is a mixture of pasture and row crops, and then at the bottom, these two more natural sites. So in general, the natural sites were less impacted by pesticides than sites with human disturbance in them. The Maumee wins or loses, however you want to look at that, in basically every category that we can come up with. So on the left-hand side of this panel, you're seeing kind of a ranking system. Blue is bad. So in this case, Maumee wins in all of these scenarios. A total of 72 unique compounds were detected in the Maumee. I had a maximum of 58 compounds detected in a single sample. But I want to point out that both the urban landscapes, the crop landscapes, and the mixed agricultural landscapes all had a lot of pesticides. And you'll see a little bit later how these relationships hold up when we consider kind of the proportion of disturbed land. And again, few compounds were detected at the wetland and forested sites. 6 in the Bad River was the lowest site. Which pesticides are present? So now we're starting to get towards prioritization. There's a lot out there. Which ones are the most important? This is a hard figure to read, and that's kind of the point because, again, mixtures of compounds, there's a lot out there. All you need to know is that red bar, this is total number of samples where the compound was detected. If it's gray, we didn't detect it. If it's red, we did. The herbicides are on the top. The insecticides, which you are all interested in, are down here in the middle. There we go. And then fungicides down here at the bottom. A lot of herbicides were detected in a lot of places, things like metolachlor, atrazine, acetochlor, but we have our favorites down here, imidacloprid, clothianidin, a large number of detects across those 16 sites. Atrazine and 2 of its transformation products were kind of the most detected. They were present in 75% of samples at all sites, even those natural sites. Neonics, I already said the imidacloprid and the clothianidin were the most frequently detected insecticides in 44% of samples. Okay, other presenters have asked the same question. Okay, they're there, do they matter to the aquatic organisms? we've already talked about this, but basically we're putting the concentrations that we measure in the stream relative to these two values that I talked about, the gold standard aquatic life benchmarks and these ToxCast values, which are more like cellular molecular measurements. We used a conservative threshold. So we, divide that 1 by 10 as sort of like a protective measure. And so we're saying anything above these thresholds we count as an exceedance in this study for screening for pesticides of concern. So do those concentrations matter to aquatic life? The answer is yes, it seems. Many times the concentrations are exceeding these benchmark values. To orient you, if you want to look at our gold standard information, our aquatic life benchmark exceedances, you can look on the right. If you want to look at this other source of information that can be used as a screening tool, you can look at the left. They kind of tell the same story. Half of our sites are potentially negatively impacted by pesticides year-round. So that means every month of the year, at least half of the sites are experiencing some sort of water quality benchmark exceedance. And in any given month, 5 or more chemicals are contributing to those exceedances. So again, mixtures, think multiple stressors, 5 or more compounds in each of these months. Okay, back to this question of who the biggest offenders are in terms of which pesticides are causing these threshold exceedances. I'm showing you the same figure I showed you before with a list of a bunch of the compounds that we detected, but now I've color-coded those bars based on the concentration that we measured and kind of whether they exceed those thresholds or not. So if it's red or dark red, that means it exceeded those thresholds. And so you can, you look up here, metallochlor was our kind of our herbicide that had the most exceedances and so on. And you can look down here and look at your neonics. Imidacloprid had a lot of exceedances, 44% of the samples. And all of the times that we had detected imidacloprid, it was above a threshold, which probably has something to do with detection thresholds and how toxic it is. But and then you can see, some of the herbicides that were frequently exceeded those thresholds. This is a lot of information at a lot of sites, a lot of time periods. And so we used a bunch of metrics to kind of start to boil down what compounds are priority, what sites are priority. And so I want to point out one thing, which is here you're seeing these dark gray bars, and that means it was detected, but we have no toxicity information for that compound. This happens with a lot of the transformation or degradation products for these pesticides. And so, maybe this is something to be concerned about. We don't know anything about these compounds. So, we might want to prioritize it just because we don't have the information and say, hey, why don't we follow up on some of these? So that's what we did. This figure is kind of complicated, so I won't talk a lot about it, but basically we used some occurrence metrics. So if it was present and we don't know anything about it, let's prioritize it. We used those aquatic life benchmarks and those ToxCast benchmarks. We combined it all, and we said if it's prioritized based on any of these metrics, it's going to make it into our list. And so we identified these 16 compounds that you can see up here as being pesticides of concern in the Great Lakes Basin. And I'm showing you the rules for how it got in or not. And 2 of the neonics were prioritized in this study, imidacloprid and clothianidin. This figure is showing you the potential impacts by pesticides as related to kind of that land use indicator. But here I'm not just showing you like percent agriculture in the basin. I'm showing you actually the sum of any urban land use, pasture, or row crop agriculture. And you can see there's this relationship with our, it's called a relative hazard index, but it's basically just like an accumulation of all that information I talked about, and then I ranked the sites. So Mommy is up here as like the most impacted. But what I want to point out is just the huge range of potential impact across these sites, even at really high disturbance values. So just because you have like 75% disturbance in your watershed doesn't mean you end up like the Maumee. There's a lot of variability here that's just kind of demonstrating it's complicated out there on the landscape. There's a lot going on, and it doesn't directly translate to potential impacts to biota in the water. And this other figure is showing you the same thing. Here's the number of detected compounds as it relates to percent agriculture plus urban. So ag, certainly isn't our only problem. Which tributaries, back to this prioritization of place, and I'm almost done. I won't walk you through this figure. Again, the Maumee won in a lot of senses. It loses in a lot of senses. And I'm just showing you the Wisconsin sites in particular. Blue tends to be more impacted than the lighter yellow colors. And you can see, still some impact at these low disturbance sites. So in conclusion, there's high potential for biological impact of these compounds in tributaries of the Great Lakes. This potential impact increases with disturbance in the watershed. They're having a year-round impact, and neonics are only part of the story. Thanks.
Good morning. I guess I might have the unique distinction to be the person to come travel the farthest. I am here visiting you from Montana. Most of the work I'll be sharing with you, I did while I was a resident of Colorado. And with some trepidation, I'll let you know that I am an alumni of another Big Ten university that just whooped you guys up on the football field. So don't use that against me. But we do have one thing in common, and that is a concern about neonicotinoids in the environment. If you have any questions or want more detail about this, there's a couple manuscripts I'll have in here to go peek at. This one here is probably why I got invited to talk here. So I'd encourage you to go take a look at that. Trying. Oh, there we go. So ecologists have long been concerned about what are the big drivers of change in the world. And in the millennial assessment back in 2005, they went out and quantified all the big forcings that could change the health of our global ecosystem. And they're really obsessed with things like nutrients and land use change for good reason. Oh, wrong direction. But largely, they ignore synthetic chemicals. And some of my colleagues have documented this in a not so recent article where they compared synthetic chemical production and diversity and use in time and showed that they're changing at the same rates as land use change and carbon dioxide emissions. But rarely do we ever really pull that into the global context of like, how are we changing our environment on a global scale? And one of the things I'd like to point out here is that while pesticide use has been going up quite steadily for a long time, in the United States, there's a good news here. We have regulations. We make choices about how we use pesticides, and we often change our behaviors. And sort of a good news, bad news story associated with that is that one of the choices that was made back in the 70s and 80s with like OPs and organochlorines were really causing a lot of risk to wildlife and birds. As you can see here, my colleagues from Germany who really like to scoop USGS data and publish it before we can, they showed that those risks were higher in the past and they declined in time. And that is, as you've heard in many of these other talks, a direct result of transitioning towards pyrethroids, fipronils, and neonicotinoids, which are more specific, more direct, like more specifically applied in the environment, or at least before 2015 they were. But because of their specificity and their increased toxicity, they are pose greater risks to aquatic insects, pollinators, and to terrestrial arthropods by design, right? They're trying to control terrestrial arthropods. So this change has happened in terms of risk, and as Samantha just pointed out, this is just applied pesticides to the landscape. It doesn't mean it actually got into the streams. So we don't know to what degree pesticides are having an impact on the stream ecosystems. There are two agencies in the federal government that study streams at a big scale, and one of them is the Environmental Protection Agency, and they do a lot of work probabilistically sampling streams of the continental United States to try to tell you what is at issue in these streams. And in one of their recent assessments, they found that 46% of the streams in the continental United States were degraded. Their biological condition was below performing standards set by the Clean Water Act. And the reasons for that were a bunch of the traditional metrics that stream ecologists tend to focus on, nutrients, sedimentation, and stuff like that. But they don't assess synthetic chemicals at all. And that's where the other agency has historically come in to place. The U.S. Geological Survey has done these NAQUA studies, these national surveillance programs where we go out and measure everything under the moon. And we do so in a way to try to understand causal linkages between changes in land use, water quality, habitat degradation, so that we can figure out what are the tools, the knobs that we can turn to try to change management to improve stream quality. And what I'm going to be sharing with you is results from what we call the Regional Stream Quality Assessment, which is Cycle 3 NAQUA. And we went out for 5 years and sampled about 100 rivers in each of these ecoregions. And then I'll be presenting to you a lot of the outcomes from that research. Why study different ecoregions? Well, because land use patterns change, right? And you'll note that the Midwest, as many of you know, is dominated by agriculture, but much of the Southeast and California and the other ecoregions have quite a bit more urban land use conditions in there. So if we're going to really understand how land use is changing and impacting aquatic ecosystems, we've got to study it at a large scale. And then it also helps us address the significance of our findings from each individual regional assessment. So when we show up, we're totally the hit and run scientists. We show up in a panel van, 14 to 20 people, and we spend a day at a site or maybe two sites. And we get there and we measure several hundred pesticides in water. We measure pesticides in sediment. We try to use the most modern methods that we have access to, but as pesticide use and behaviors change in time, there are new compounds getting on the market over decades, and over every year we fall behind. So it's tough keeping up with that. But then we also do a really comprehensive physical habitat assessment, and we collect bugs, fish, and algae using quantitative ecological metrics. We take that data home and we crunch it. We analyze it until the cows come home to try to learn what's happening in there. One of the outcomes from this study that I really wanted to make sure, looking at the speakers list from this morning, I put this slide in there just to emphasize that one of the outcomes of this study was that even in the Midwest here, that was dominated by agricultural land uses, we found very strong urban gradient. There's an urban pesticide signature. There's 16 pesticides that are unique, almost unique in terms of their frequency of detection and concentrations that you find in urban and exurban environments. And this is a paper that I wrote, and it shows you here. We got the land use linked to a pyrethroid here, and you can see urban and ag use are both affecting the concentration of bifenthrin in these ecosystems. So this is not just an ag issue. This is pesticides in the environment is also a growing urban issue. Another outcome was that we put all of this data, I think it was over 650 metrics that we measured, including all those over maybe 300 different measures of pesticide toxicity, into some machine learning models and crunched it and asked, what are the most important predictors of two ecological metrics? And look what came out to be one of the most important ones, but contaminants. Whoops, and I just stole my message. Those contaminants were dominated by pesticides. And it was in every single ecoregion that contaminants was a really strong predictor of ecological health. So pretty good correlative assessment, first cut screen of the data. So then we dug a little deeper and figured out, okay, what contaminants are we really concerned about? And so we were able to look at the most frequently detected insecticides in the United States. And it's a common theme that you're starting to hear. Fipronils, neonicotinoids are some of the most commonly detected insecticides in surface waters. So we found the roughly 500 streams that we sampled, 32% of them had at least one detection of imidacloprid. But as I said, we were always struggling to keep up with all the different pesticides being used and applied and detected in the ecosystem. So when we measured these pesticides, we had exactly one of the 7 commercially available neonicotinoids in our method. So this is a gross underestimate of the frequency of neonicotinoids in the continental United States. By the time we got to California, our last regional ecological assessment, we were able to add the other 6 commercially available neonicotinoids. And as most of you are familiar, California is sort of the breadbasket of the United States. Quite a bit of vegetables come from California. But they are also exceedingly regulated. They have their own pesticide regulatory authority. They even regulate pesticides in urban environments in California. And so I would suggest that this is not like the worst case scenario ecoregion that we studied. So what did we see? Pesticides, very similar to Samantha's talk, in 67% of the sample locations we sampled in California, including reference sites. And the concentrations exceeded EPA aquatic life benchmarks by over a factor of 7. So it seems like streams are commonly have exceedingly chronic toxicity levels of pesticide mixtures. Most of that toxicity is dominated by neonicotinoids, and we note that whilst not the most frequently detected neonicotinoids, clothianidin and imidacloprid, according to these EPA benchmarks, were the most toxic. So we got curious, how toxic are they? Now, one of the real issues is we get these studies, we do field assessments, we measured, I think 182 different insecticides were detected in the streams that we sampled. I think the average number of insecticides or pesticides was like in the 50s in every sample we collected. So in stream ecosystems, we have multiple pesticides and other contaminants. We have natural covariates. When you're trying to study biological assessment and you're working at a very large scale, bugs and fish, they change their range. There's biogeography, so you got to control for that. You got to deal with other limiting factors like contaminants that we're not talking about today, PCBs and stuff, PAHs and stuff, and heavy metals. And then, as is frequently occurring theme in these talks today, we don't really understand the interactive effects of these mixtures, and we know it's a foregone conclusion. Most streams are mixtures of pesticides at this point. So how do we know which one is limiting that stream? One good way to get at that is to do experimentation. So you make these field observations, you draw some conclusions, and then you test to see if you can see the same thing under highly controlled environmental situations. So US EPA and lots of other agencies will do bioassays, which are really handy. They're very predictable, they're replicable, we can control the error in a precise way. They also tend to focus on organisms that are really easy to grow in a beaker or in an aquarium. So these are robust organisms. So the databases that we're performing these risk assessments on are dominated by non-insect taxa, but we're concerned about insecticides. So these benchmarks are set based on crustacea, which have a different physiology, which may not be sensitive to an insecticide. So we need to move on from this and move towards methods that we can do highly replicable, controlled study designs and measure not only aquatic insects, but also ecological communities so that we can take that data back and compare it to the field observation so we can build a better causal linkage with exposure and effect. And a good bridge between the bioassay and field observation is this thing called a mesocosm experiment, because it works at population and community levels. It's not quite as complicated as an ecosystem, but it's certainly more complicated than a cell or a beaker full of just one taxa. And because we're getting closer and closer to reality, it improves our ability to speak causally about the relationships between exposure and effect. So this is my mesocosm lab. This is what I now call the Montana Experimental Stream Observatory. It is really 36 buckets held in bait chambers where we control temperature. This is about when we do one of these experiments, about a 50 days straight, no days off. We have to go out and put trays in a river, and we colonize them out there for at least 30 days. We then have to go out and collect them. We put them in coolers and bubblers, and we drive them several hours from our reference site to my lab. We also collect all of the water to run a 30-day mesocosm experiment at one time so that there's no changes in the water quality during that 30-day exposure. We truck it, pump it into the lab, and then we do rude things. So we put all these trays of invertebrates in these different streams, and we expose them to concentrations of pesticides and trace metals. Not only do we count the number of surviving larvae in the streams, but I also count the number of insects that are emerging from these systems. And this is what a mesocosm looks like when we're breaking it down and we collect the invertebrates. And just so you guys know, these bugs tend to be happy. This is Brachycentrus americanus. Here's a Baetis. Here's some Rithrodgena mayflies. This is Arctopsyche grandis. There's real videos taken from my mesocosms during an experiment. So they're getting along just fine until they get exposed by something. So one of the things we wanted to do is say, are these aquatic life benchmarks protective of ecological communities? So here we have the number of mayflies I saw in these mesocosms plotted here. And this HC5 is basically a metric that says 95% of the organisms in the streams would be protected, right? So the EC5 that we calculated from this mesocosm experiment and using all the EPA databases is about 17 nanograms for imidacloprid, which is a little bit higher than the EPA aquatic life criterion. So it seems like maybe the EPA aquatic life criterion for imidacloprid is protective of mayflies, total mayflies here, right? And then we did the exposure with clothianidin. And here we learned something a little different. Clothianidin doesn't have as rich a database. And so what we found, I doubled the number of taxa that are in that database, and we calculated the concentration at which 95% of the organisms in that database would be protected. And that's closer to the US EPA criterion is 50 nanograms per liter, and I'm measuring that 10 is actually where we need to be to protect invertebrate communities. So to understand how mixtures affect this community of very sensitive organisms, mayflies, I took these two dose response curves and compared it to our third treatment in this experiment. So we did a treatment with imidacloprid, we did a treatment with clothianidin, and then I mixed the two together. And I'm going to do something called response addition to test the question, are mixtures of pesticides additively toxic or more than additively toxic or less than additively toxic? So we're using these dose response curves to predict the mayfly response in the mixture. I already made that point. And so when we plot the predicted number of mayflies versus what we observed, what we found is that as the concentration of exposure increased, we saw larger and larger effects. So we call this dose-dependent synergism. So the impacts that we observed in the mesocosms were stronger than expected if you just added the toxicity of the individual compounds themselves. And this is actually the weakest evidence in this work for that synergism. So then I wanted to take that information and see how it would translate to the field. So what we did is we went to the California database and we plot all of the total mayflies that we measured in the streams in coastal California, right? And behind this is the multivariate model. So I've already modeled out all the covariates, the natural land use gradients, precipitation, lat and long out of the data. And then I fit a bunch of toxicity metrics and asked the software, which is the best model? And the best model that came up to describe these mayfly responses was one that included an interaction term between clothianidin and imidacloprid. So this is a multivariate model I'm plotting in two-dimensional space. And lo and behold, we get dose response curves. And not only do we get that, but we see that the inflection point of decline is really close to that HC5 number that we calculated from our lab study, saying that this is where we should start seeing more than 5% of the taxa to fall off. And then this, I called it urinary EC50. This is where 50% of the mayflies should have been killed due to the exposure to just one of these two. And we're seeing that in the field, we're extirpating mayflies when we get to the EC50 for the individual compounds. So that is field evidence that we are seeing larger than additive responses due to mixtures to neonicotinoids. And there are also, not only are we killing individual larvae, but aquatic insects serve a very large ecological function outside of the river. We often measure them as larvae. This is what we're concerned about when we're talking about trout. But to complete their life cycle, they have to emerge from the stream. And when they emerge from the stream, they fly into the riparian zone. And riparian zones are where birders go to see birds. And there's bats, and there's spiders. There's all kinds of organisms in riparian zone. There's a reason. Not only are they finding more prey items there because the number of bugs coming out of the stream, but we now know that the aquatic insects concentrate fatty saturated acid, polysaturated fatty acids. They're more nutritious. And so there's plenty of evidence out there saying that songbirds feeding on emergent aquatic insects are way more productive than songbirds feeding on terrestrial organisms. And so there's a reason why they're coming there. So what happens when you put neonicotinoids into a stream? And I'm getting shooed. What happens when you put neonicotinoids into a stream and you quantify emergence? So this black line here is the controls, and I basically subtracted the number of control emergers from each of the doses, imidacloprid, clothianidin, and imidacloprid. And I'm not showing you the response addition plots here, but the strongest evidence for synergistic effects were actually in these emergence responses. So we've seen this with multiple contaminants that whilst you can go measure larvae in the stream, at some streams, they may not actually be emerging and completing their life cycle. And with that, just leave you with some large take-homes. We found imidacloprid to be the most detected neonicotinoid across the continental United States back in 2018. Mixtures of neonicotinoids, when we measured them, were really common, and they were dominated by clothianidin and imidacloprid, which cause synergistic effects. We also note that we measured that imidacloprid itself was a limiting factor to invertebrate communities in 4 of the 5 ecoregions we studied. And that, I stress this, neonicotinoids and pesticide use is not just an agricultural issue. It is also an issue that we control in our homes, in our cities. And so we need to broaden the conversation to account for both of those. And if you guys want any other details, you can see the manuscript. This is the one I'm mostly focused on, but this manuscript is actually a very, very comprehensive view at the total data set. So with that, I can take questions or go to the next speaker.
Going right through your stop signs, aren't we? Yeah. So for my portion of this presentation, I'm going to be talking about our investigation into the decline of burrowing mayflies on the Upper Mississippi River, and so really this culminates about 3 years of work that we've done on the Mississippi River in the Upper Mississippi River states. Yeah, so really, in a lot of ways, on the Mississippi River, I think we're incredibly lucky to have the Hexagenia burrowing mayflies. We've oftentimes referred to the Hexagenia, as the canary in the coal mine. We've lost these burrowing mayflies in certain times and places throughout history on the Upper Miss, and we've subsequently gotten them back. And so really, for that reason, they're really almost the most no-nonsense ecological indicator of health of the system. When I came to my position, my predecessor John Sullivan, advised me, keep an eye on the burrowing mayflies. They're really going to be able to tell you, kind of an early warning sign of things, happening on the Upper Miss. And so obviously, for that reason, that's something we really pay, very close attention to. And so, really, if you ever only read one book about the Mississippi River, this is the one I recommend. Immortal River, published by Cal Fremling. He was a longtime professor at Winona State University. And really this book was published at the end of his career. He spent over 50 years studying the burrowing mayflies on the Upper Miss. And really sort of was early in sort of recognizing the importance of the burrowing mayflies. And within that chapter, in his book, he mentions that the Hexagenia nymphs live in sediments where toxins accumulate. They have no tolerance for zero oxygen conditions, cannot swim long distances to escape stress, and have a relatively long life cycle. This makes them really one of the ideal sort of bioindicators, not just on the Mississippi River, but on other systems. And so, just to get into some basic, burrowing mayfly biology, there's two major species of Hexagenia. In North America, the largest hatches in North America, occur both on the Mississippi River and on Lake Erie. They'll spend 1 year, on the bottom of the river, in these U-shaped burrows. The entire life cycle occurs over no more than 2 days. And the gravid females will lay about 8,000 eggs on the bottom of the river, and the cycle essentially repeats. It's really worth noting that one of these individual adult Hexagenia is a little over 100 calories. So if you do the math on these systems like Lake Erie and the Upper Miss, we're talking trillions of calories available to birds and fish too. I grew up on the Mississippi River, where many times when the hex hatches were going on, we wouldn't even bother fishing for a week. The fish would be satiated. So really, the caloric importance of these to this system really can't be underestimated. And so, we really have the two species of Hexagenia on the Miss. We have Limbata, hatches earlier in the season, lighter yellowish in color. Bilineata hatches later in the season, darker color, and generally more abundant overall. And these are pictures, that I've formerly used in presentations, mostly to talk about how awesome we were and, brought these mayflies back too. But, really, piles of burrowing mayflies at Lock and Dam 3, almost up to the door handle. These look like summer storms, but these are actually Hexagenia hatches on the Mississippi River. They actually look like intense summer storms, but these are Hexagenia hatches on the river. And so, really, I've tended to use these less and less. It's not as if we don't have these hatches anymore, but they're more subdued. They're farther and fewer between. There were many of us working on this system. Have noticed over the last 15 years that these hatches are becoming more subdued. And so, really with these anecdotal observations that we had, every once in a while, a paper will come out. There'll be a bit of a wake-up call to the community. And this Philip Stepanian paper, basically what this did is it analyzed readily available weather radar data, both on Lake Erie and the Upper Miss. And really documented it, in recent years, really steep declines in these burrowing mayflies, across these two widely spaced systems. So really, when you have this occurring over broad geographies to this, this was a big wake up call for many of us. This isn't a localized decline. This is widespread, subcontinental declines. If you look at the mayfly abundance on Lake Erie, looking pretty good till about 1960. We lose them. The Clean Water Act and many of the pollution reduction activities that followed. We brought them back. Here we're feeling pretty good about ourselves too, and then this tailing off in both systems. And so, Yeah, and so, on the Mississippi, we're awfully fond of Mark Twain quotes, or this reminds me, of the Mark Twain quote where, history doesn't repeat itself, but it often rhymes. And, not surprisingly, after the Stepanian paper came out, National Geographic, seized on this. Many of us on the Miss have taken, Nat Geo photographers out to film these hatches on the upper Miss. This is a globally significant biological phenomenon that occurs on the upper Miss every year. And, even the local press, the Milwaukee Journal Sentinel, did a really nice article on the mayfly decline this past summer. And so, really when the Stepanian paper came out, I was talking to my colleague Steve Delane with Minnesota DNR, and really over that same period of years as that radar paper, Steve Delane and his group with Minnesota DNR up on Lake Pepin, we're doing a benthic dredge program. The ingenuity of these river biologists is incredible. They essentially modified gold mining equipment, to be able to vacuum the bottom of the river to collect Hexagenia mayfly. You essentially have to get 25 grams of Hexagenia to be able to run body burden analysis for contaminants. And they developed this method. And so really, while this method wasn't designed to detect trends, his group reported that it was over that period of years it was becoming more and more difficult to collect the required amount of mayflies to run this tissue analysis. So again, sort of another line of evidence that got us thinking, something was going on. And so, really, over the next year, myself and colleagues from Minnesota, Iowa, Illinois, and Missouri, we worked really hard, and we put together a proposal to reinstate the benthic invertebrate sampling program on the Upper Miss. And so the long-term resource monitoring program on the Miss is a series of 6 field stations in these upper 5 river states. And this benthic dredge program, using a Ponar dredge, allows us to estimate benthic invertebrate densities on the bottom of the river. And, it was mothballed in 2004, due to budgetary constraints, and we were essentially able to bring that back for a 3-year period. And, and this data, is year 1 of a 3-year period, but just an analysis of medians, this apples to apples statistical comparison, showing about a 45% decline, which is generally, in line with what some of the radar estimates are showing, kind of a 50-ish% decline in recent years on the Upper Miss. And so, really what's changed over that 19-year period, one of the more obvious things that's changed is just the amount of water we're dealing with on the Miss. This is mean annual discharge at the Mississippi River gauge at Winona, 1920s to present. In 1929 to 1980, we never had a year with mean annual discharge above 45,000 cubic feet per second. Conversely, we've crossed that threshold 12 times since 1980, and 7 of those have been since 2011. So the prophecy of warmer and wetter, it's here. And so, really, that's a lot of change over a relatively short period of time. And really one thing we're seeing, the bottom of the river with these benthic dredge surveys, when we're collecting tissue for these mayfly samples, a decent amount of the substrate in the lower thirds of the Mississippi River pools, where we expect to see Hexagenia, is now spent gastropod and zebra mussel shell. So again, it's not beyond thinking about the just simple habitat shifts. Could be at least part of what's going on, relative to this mayfly decline on the Miss. And the other thing, really good presentations by Samatha and Travis. A lot of these current use pesticide papers, dealing specifically with the neonics, and their effects on aquatic invertebrates. These papers, the last 10 years, very, very high number of papers, really high quality papers documenting this. And even, as it's been mentioned, it's not only just the out and out lethal effects. This is a paper that was interesting to us on the Miss, looking at both lethal and non-lethal effects of the neonics, specifically on the Hexagenia mayflies that we're interested in too. And so really, it's not only just like these lethal effects, but behavior and mobility effects. This panel, off to the right, is showing, at certain concentrations of neonics, the Hexagenia will become agitated to the point where they'll leave the burrows. anybody that knows anything about the Mississippi River, if you're leaving the boroughs on a pretty regular basis, you're putting yourself at heavy predation risk, just based on the fish densities out there. So there's different ways to die out there, I would say. And so, really, in 2021, Mike Miller and myself, we set out to collect baseline data within the state of Wisconsin, to basically, figure out where we were at from a baseline standpoint, and really compare the waters of the state, to some of these EPA benchmarks, some of the various published papers that were out there too. And so, we kind of took a divide and conquer strategy. And so for my portion of this, I sampled the 10 major tributaries on the upper Miss from the St. Croix River down through the Platte River, and then the 10 lock and dams within Wisconsin border waters, Lock and Dam 3 through Lock and Dam 11. And so, the tributaries on the x-axis will be the St. Croix through the Platte, and then the lock and dams north through south. And so really all told, we detected neonics at 95% of our sampling sites. There was 2 instances on the St. Croix River and 1 on the Chippewa, where we didn't detect any of these 5 major neonics. Not surprising, these are more forested watersheds in the northern portion of the state. And, we also compared our data to some of these published papers. This dashed reddish brown line is essentially the long-term chronic concentration from a recent review paper. In this case, in this large rivers data set, we didn't have any of those sites rising to that level. But this blue dash line is from Travis's paper that he just referenced. So this dash line is 10 nanograms per liter clothianidin, which is essentially the hazard concentration for 5% of species. And interestingly, the test mayfly ephemerella was most sensitive to clothianidin. And so really amongst this large river data set, a little over 8% of our samples were above that hazard concentration for 5% of species. And so really for Mike's portion of this, I kind of took the large rivers piece of this, and Mike, kind of dealt with sort of the small and medium-sized streams within Wisconsin. This is in my mind probably the most compelling data of this too. And so, what Mike did is he stratified land use within Wisconsin into 4 groups. The urban streams, comprised largely in the southeastern portion of the state. The vegetable cropping portion of the state comprised largely in the central sands. The row cropping areas of the state scattered throughout the state. And these blue sampling sites are essentially everything else other than urban vegetable crops and row crops. And these data, extremely eye-opening. So for these box plots, the X here is the sample mean, and so really in the vegetable cropping portion of the state, the sample mean, is above even acute toxicity from review papers that are out there. And in the urban and row crop portions of the state, the sample mean, at or above chronic toxicity. So again, a little bit different than the large rivers in the state, but very eye-opening data. And so Mike, he also applied this small and medium-sized streams data set to publish the EPA aquatic life benchmarks, freshwater chronic benchmarks. And so for clothianidin, he observed, over 80% of the sites in the vegetable cropping portion of the state above that clothianidin metric, EPA aquatic life benchmark. The corn soy, about 35% of the sites above that benchmark, the EPA benchmark. And then for imidacloprid, 60% of the sites in the vegetable cropping portion of the state. And then the corn, soy, and urban, about 25% of the sampling sites above those EPA benchmarks. So again, really eye-opening numbers, on sort of this fairly comprehensive statewide survey. And so, really, after that initial round of data, Mike and I, we really knew that, this was sort of an area that needed, further exploration. And so really what we did is we paired up with the EPA research lab, Office of Research Lab in Athens, Georgia. And what I did for this, I went out and sampled, again, those 10 major tributaries and 10 lock and dams on the upper Mississippi River, and really intense chemical screens. I think 9 different chemical schedules at these sites too. And these are the top 31 compounds detected. And Travis talking about where many times it's not only these compounds one at a time. It's the mixtures. There can be sort of an effects stacking that goes on. But just to look at some of the mixtures that we're seeing on these tributary rivers, and rivers on the upper Miss, and really of the neonics, clothianidin, imidacloprid, thiamethoxam, 3 of the top 13 were neonics. And also bringing in, Samatha Oliver, her chemical prioritization paper. 7 of these 31 compounds were compounds that were pulled out in Samantha's chemical prioritization paper of Great Lakes tributaries. And so, okay. Yeah, and so adding, in June of 22, we also went out and collected some sediment samples on Pool 8 up by Elma, Wisconsin, and Pool 8 near La Crosse, and Pool 4 up by Elma. Again, these burrowing mayflies live down in the sediments, and we wanted to get a sense of what compounds they were being exposed to in the sediments too. And so really, this plot is just showing the sum of all detections. These are the top 70 compounds detected within the sediments. And again, three of the neonics, at least in the top half or so amongst those detections too. But again, from a mixture standpoint, there's a lot of mixtures, that we're finding amongst compounds, in the sediments of the upper Miss. And so, really progressively, as this project went, we eventually added layers to this. And so really as part of getting the invertebrate protocols back on the upper Miss, there was an aspect of this project where we were in these 6 field stations. We went out and collected paired water, sediment, and mayfly tissue. So we used that benthic dredge method at these 6 field stations throughout the upper Midwest and in these 5 upper river states. And we were able to essentially run how much of the pesticides we were picking up within that mayfly tissue. And this chemical schedule is focusing largely on the neonics too. And so, really these sites are largely north to south. So here's Minnesota, Wisconsin, Iowa, Illinois down by St. Louis, Missouri, and then the LaGrange Pool on the Illinois River. And so, really all told, within the mayfly tissue, we're picking up clothianidin, in 100% of mayfly tissue samples amongst these samples in the upper Midwest. Picking up imidacloprid in roughly 59% of samples of mayfly tissue in the upper Midwest. So again, really eye-opening information. I don't think we really expected this going into that. And then we also ran, one other chemical screen of that mayfly tissue from these upper 5 Mississippi River states. And so this is the multi-residue pesticide run. And these are the 30 compounds that we detected within that mayfly tissue. There was 32 compounds that were not detected in any sample too. And so, again, from a mixture standpoint, a lot of chemicals being picked up, within the mayfly tissue itself. And certain compounds like atrazine, were picked up in 100% of our samples across the Midwest. With a decent amount of range too. So again, from that mixture standpoint, and really what we're seeing, this has been incredibly eye-opening to many of us working on the upper Mississippi River. And so, really, as we've sort of progressively added layers to this, in 2024, we again sort of teamed up with that EPA research lab too. And so, in 24, we're collecting paired water, sediment, mayfly tissue, and also adding what's called metabolomics. And so really what this is, is we'll go out and collect mayflies. There's an extraction process and essentially a spike reading exercise of metabolites. These are essentially, biochemical markers of stress, related to pesticide exposure. It could be other exposures, thermal thermal stress, oxygen stress too. But again, we're really hopeful that this paired data set, with this range of conditions, throughout the upper Midwest, is really going to be able to open things up. This is sort of a new and emerging line of science that we think is going to be really promising. Really none of this data is in hand yet, but it's we think this is going to be really promising. Yeah, and in conclusion, I would just like to thank some of the people that worked on this. It's some of these benthic dredge crews on the river. I owe these guys beer basically for the rest of my life, and then just everyone from the LTRM that worked to collect these data. Yeah, so that's it for me.
Yep, so Sam and Travis, if you could come join us, and Shawn up at the front. So just a reminder then, lunch is going to be served right outside here to the left. You'll get your lunch there, and you may have to come sit in here if there's any available seating out there in the atrium. And just to let you know, they may be, during preparation, may be opening up this side, so I didn't want to think that was time for lunch, but just to let you know. So we'll start with questions then in the room. I'm sorry, did you have? Michael, I seen you pointing.
I got two online.
Go ahead.
Okay, so the first one is for doctors Oliver, Romano, and Schmidt. Are you accounting for duration or average concentration over time, comparing chronic EPA benchmarks to chronic concentrations or acute to acute concentrations?
I can say in our study we used kind of the lowest benchmark that was reported for EPA, so you can consider our reporting conservative. But again, we were measuring through time, so we're definitely considering kind of concentrations over the long term.
In our national scale surveillance, we used both acute and chronic benchmarks. And then when we went through each ecoregion, we found different insecticides were important. So we ran mesocosm experiments. And all those mesocosm experiments were run on a 30-day time cycle, which is about what the chronic criterion are supposed to be targeting, but virtually nobody runs tests that long. So yeah, our effort was specifically to try to get at the longer term effects of having insecticides in rivers.
Thank you. The second question was for all speakers. Did your program include analysis of matrix spikes for your water samples? And if so, did you ever find evidence of matrix effects on the concentrations of neonics?
Yes, the long answer. This we could have a whole day session on all the complicated matrix effects and the changes in detection limits and the changing in method detections and all of that. But in all our national surveillance, we censored all of the data, and how we address that can be directly seen in our supplementary information in all of our interpretive products. So that's all plain language, publicly available information.
Great. For questions in the room, Michael has the microphone. If you'd like to raise your hand, please.
It seemed like a lot of the data presented today really focused on imidacloprid and clothianidin. Curious on thoughts of why thiamethoxam seems to be maybe less prevalent in the data sets, or if there's considerations there on which type of neonic might have the biggest impact.
Yeah, it definitely seems, like clothianidin and imidacloprid are the two, they keep pulling out of paper after paper after paper. It's like I think you just, even just the evidence presented in this session, those are
too. And I think the breakdown of thiamethoxam to clothianidin is a big piece of that. So I mean, I think it's sort of a breakdown detection piece there as well.
Some of it has to do with the correlations from field data. If we don't see strong correlations from field data, then we tend not to focus on it as much. And the course cut on EPA benchmarks are they're relatively less toxic than the other things, and then they're just so frequently detected. Part of it was that imidacloprid was like the first one used, and then clothianidin and then the others started to become on the market. So imidacloprid got a lot of attention early, and the other ones are getting more attention through time.
You've gone over a lot of data this morning. I'm curious where you're headed next with your research. I've seen a lot of head nodding to the fact that there's a lot more that needs to, a lot more data that needs to be collected, a lot more stream segments to be looked at, all of that. Where would you like to take your research next to answer some of those big untold questions?
I can answer from the GLRI perspective. My colleague Steve Corsi is in the room, and they're right now investigating how pesticides behave in streams as it relates to storms or hydrologic events. So I showed, we took monthly samples, but we did not target high or low, stream flow conditions. It was just kind of like when we were there, we took a sample. And our samples were pretty representative of the general hydrology. But you can think of all of the things that happen if you have a really big rain event and what that might mean, say at the field scale or relative to when the pesticide was applied. So they're looking at like variability over the hydrograph of pesticides in streams.
Yeah, and I think for me, that metabolomics data set, we have paired water, sediment, mayfly tissue, and those metabolite markers. I mean, I think that's going to really open things up. So it's, again, sometimes it's a waiting game on these data, but I think that data set is going to really open some things up for us.
When I started with the NAQUA team, like 2012, 2013, the question really was, would we see that pesticides have an effect on aquatic ecosystems at the regional scale? We really didn't know the answer to that question. Then come 2018, 2020, we have an answer to that question. In most ecoregions that we studied, there's a clear link. So those are in small, wadable streams. And if you look at these pesticide application maps, some of these insecticides are being applied at very large spatial scales. So we're now asking questions that I think some of your speakers here in the room are leading the way on is, could these insecticides be impacting large rivers? And so we're right now doing some work looking at the concentrations and trends and loads in the Mississippi River for a suite of pesticides. One data gap, and it's not related with neonicotinoids, but is concerning. Throughout all of our studies, fungicides are often seen at the highest concentrations and highest frequency detected in these samples. And some of those fungicides have chemical properties that suggest that they could be bioaccumulative. They could also be potentially toxic to non-primary producers. So we just started with, I just ran a mesocosm experiment this summer, looking at the effect of 3 of the most common fungicides on algal productivity as a first start. But ultimately, we'd like to know if they are possibly having impacts on aquatic insects.
Question?
Yeah, this is for Travis. Your metric focused on the protecting 95% of the mayflies, but you showed in your mesocosm you had caddisflies, and I assume you had other things as well. So is there a relative ranking of sensitivity to the things you're looking at between these different taxa? Are mayflies the most sensitive group, or plecopterans, or other groups?
Yeah, for the longest time, I thought I'd see plecopterans being stoneflies would be really important. But man, they're just hard to get in good numbers in my mesocosm. They're tough to work with. They might be a little too sensitive. The big surprise, chironomids are really sensitive. They're not the most sensitive, but as stream ecologists, we're usually like, oh, those are the weedy species. And some of them are. So there are some chironomids that can withstand a lot of, rude things being added to the water. But mayflies, I've done, 13 different contaminant mixture experiments with trace metals, pesticides, coal tar sealant. It's always a mayfly. They are always sensitive. In fact, we've compiled a metric that we call sensitive mayflies metric. That is always super sensitive. Rithrodgena, Epioris, Ephemerella infrequens in my mesocosm tests in Colorado. Now I'm in Montana, so we got different mayflies, and we'll keep chasing them.
Is there a question online?
Yeah, we have one more online. This is for Shawn. How did your 2023 surface water pesticide data from Athens, Georgia compare to other states, not just Mississippi River water quality data? And do you think that these concentrations found in nearby states could have detrimental effects on this genus throughout US EPA Region 5?
Yeah. In terms of, I think the one thing that was surprising about that data set is, some of the gradients that we saw within the Midwest. Like I had expected, there's sort of an expected like water quality gradient that we'll see, amongst those Mississippi River pools where, pool 4, that Minnesota field station will be, a lot of water coming out of southern Minnesota. The Minnesota River, I think a lot of people are familiar, it's a highly impacted river where, for many water quality metrics, the pool 4 will be, quite similar to those southern field stations, kind of down by St. Louis too. But it was quite obvious that if you get into central Illinois, that these concentrations were just far higher in that central corn belt. So it's it seems like there are distinct differences in use patterns based on what we're seeing.
Question?
I was wondering, have you measured neonics in wetland habitat or areas where there's more microbial activity? My rough understanding is that microbes can degrade neonics, so I was just, I'm looking ahead of, degradation and things like that, so I was just wondering, have you noticed that you're maybe above and below wetlands or areas with more microbial activity? Have you looked at that?
Yeah, this is maybe more up Mike Miller's alley too, but we've sort of talked about the next year or two, and that's one thing, that's sort of on our to-do list is to kind of look at, wetlands across different land use gradients. So yeah, that might really be the next thing we tackle in Wisconsin.
Look at Christy Morrissey's work out of Canada, Saskatoon area. She's studied neonicotinoids in wetlands around canola oil fields, and they are fantastically high concentrations, higher than anything we've shown in any of the data plots you've seen here, by orders of magnitude.
Okay, one question. First off, great talk, all three of you. Thanks for the information. Question for Shawn. So now that you're seeing basically concentrations pretty much everywhere you look, and a good portion of those above water quality benchmarks, does DNR, or what's the path forward at DNR to try and, modify regulation of these, or do you know yet?
I think we're in such the early stages too. I think a forum like this is, goes a long way to, kind of getting us to that, the thought process related to that too. I think we do have plans this next year to, kind of add, what we see to be like sort of missing pieces of data, like the wetlands piece, things like that too. But it's, I just think it's in the early innings of kind of, on the regulatory. I know that's going to be talked about this afternoon too, but I definitely think we're in the early innings of that.
Question?
Hi, I'm Jenny. I'm from DNR. I work with Shawn and Mike and various people, and there's some collaboration to come with DACCAP. I've been thinking a lot about chironomids, just kind of looping back to that a little bit. And I'm wondering, from the expertise in the room and at the panel, how do chironomids compare with like the caloric importance as far as their emergence goes relative to the mayflies that a couple people talked about?
I would guess. I mean, guessing. I don't know. But I do study a lot of riparian consumers now as indicators of water quality. Spiders, songbirds, tree swallows. It depends on what organism you're talking about. Spiders don't like to have things with big wings in their nets, so they almost target midges. So if you're looking at spiders as a riparian consumer, like they're I think often like dominated by midge food. I think they're super important in terms of numbers. Birds, they're going to capture more large conspicuous things that don't fly very well, like a mayfly.
Thanks.
One or two more, or do you want to close it up? No, if we have time, another question, there's one over there.
You guys are always on the opposite side.
We'll just keep you running back and forth, Michael. So.
Bad shoes I chose today. The microphone is already turned on.
Yeah, I'm not certain who this is a question for. Maybe you guys would like to bounce it around, but I'm having a difficult time reconciling the first session of the morning about the prevalence of neonics on seeds and the ag setting with some of the information we just heard about neonics and other pesticides in urban settings. And so does that mean that neonics are applied in landscaping products in urban settings, or would it be pesticides like communities that are going after bed bugs? Or just like in the grander scheme, I had the impression earlier in the day that, okay, it's all about ag. And then I've kind of lost track of that bigger picture.
I think it's all of the above. You can certainly have long-range transport of compounds, say, in the atmosphere that could then be landing in any environment, and then you measure it in the water. So there's certainly that. But I think Travis talked about a study. The urban landscape has a really specific pesticide signature, which suggests that there's a special way that urban environments are using pesticides that's distinct from agricultural areas. So I think it's all of the above, but I think urban areas are distinct in what they're using and how they're using it, is my interpretation.
Yeah, I mean, both. Who's got a cat or a dog at home? How many people treat their animal for fleas and ticks? We're using some of these insecticides on those animals. We're using them to treat fire ants, other kinds of ants, building materials. Look at California. They have regulations on how you apply pesticides to building materials, but also it's standard code in many states to treat building materials with pesticides. And then, your garden, you go to a garden supply store, you buy plants. They've been treated before you bought them, unless they're telling you they've not been treated. There's a unique urban use signal. It probably has less to do with seed coats, and that's why we see the unique signatures.
Any other question? Yes.
Turned on, okay? Yep.
This is a follow-up to Samantha, to the previous question. You combined urban and ag together to present the outcomes. Did you look at them separately? Because this morning's talks, one of this morning's talks, I think, showed less of a response in urban settings than in ag settings. So I wonder if you can comment on that.
There's a little bit of that in the paper, kind of pointing out the differences between kind of the urban and ag landscapes, but we were really interested in just kind of like really high level, where should we be prioritizing pesticide mitigation efforts? And so you could see in those like disturbance plots, the urban sites. So, you know, the metric was a combination of ag and urban, but really those urban sites are mostly just urban. And in terms of like high level potential impacts, you know, they were right there in the mix. Now those are different compounds potentially impacting biota pretty differently. So there's lots of nuance there that we didn't get into, but certainly high level, like if you have 75% urban area or 75% row crop agriculture, they can kind of be in the same mix in terms of potential impacts from pesticides.
Great. One more question over here.
I won't make you walk. With all the chronic exposure to all these invertebrates and things in the water, are you seeing any resistance buildup? I know it's a pretty hot topic among pesticide use.
He's asking if we're seeing pesticide resistance at a large scale. I don't study that specifically, but I know of a couple examples. So the Colorado potato beetle has evolved some insecticide resistance recently, and so some of these RNAi technologies are being built like specifically to go after that organism. But in an aquatic ecosystem, Hyela azteca is often a lab rat species that compliance laboratories have. And if you go on the landscape and you look at their genetics, you can see that many populations in areas that use pyrethroids have evolved tolerances to pyrethroids. So it's happening in target and non-target species on the landscape.
Okay, we are at time now, so again, is there a question? Oh, great. So yeah, just take this opportunity again to network and to ask our panelists any questions you have during lunch or during the break time. Again, while they're here is the time to ask the questions. We'll be back at 1 o'clock, and any updates then?
Quick update on lunch. All the food is behind me, behind this wall, but they've decided instead of opening it, it would be preferred that everybody exit this way because you do need to show your ticket to get into the food area. Thank you.
Okay, thanks everyone.
Recording stopped.